Hello students, in today's session we'll discuss blastula and implantation. Blastula and implantation. You already know that immediately after fertilization or after fertilization, the diploid zygote enters into the three developmental stages: cleavage, blastula and gastrula. So you already know that cleavage is a repeated mitotic division of diploid zygote that results in multicellular morula. And you remember one thing, a morula is an intermediate stage between cleavage and blastula. During the process of blastulation or the formation of blastula, the multicellular morula is transformed into a single layer blastula by the process called blastulation. So let us discuss. So blastulation. So multicellular morula is transformed into a single layer blastula. After the formation of morula, the cell division temporarily stops. And the cells start to move towards periphery, or the rearrangement of blastomeres takes place, and as a result of this the cells arranged in a single layer, and that we called as a blastoderm. As a result of the movement of cells or rearrangement of cell, a cavity is formed, a central fluid filled cavity is formed, and this cavity we called as a blastocele cavity, and this process we called as a blastulation. So in case of human being, the structure is called blastocyst or blastodermic vesicle or blastula. When you look at the structure of blastula, it is covered by outer cells called tropoblast cells or tropoectoderm. Okay. Tropo means food, blasta means layer. And the second one is inner cell mass or embryoblast or embryonal lobe. A cavity is called blastocele cavity or segmentation cavity, and cavity is filled with the uterine fluid. And blastula is differentiated into two poles. One is animal pole, another one is a vegetal pole. Animal pole is commonly known as a embryonic pole, whereas a vegetal pole is known as a embryonic pole. So when you look at the structure, so towards the animal pole you find embryoblast cells. Towards the vegetal pole, embryoblast cells or inner cell mass cells are absent. So based on this, we can identify the polarity in case of blastocyst. Okay. Next one, the fate of structure of blastula. I told you the blastocyst is made up of two types of cell: inner cell mass, and the inner cell mass which future develops into embryo, and tropoblast cells. One, it helps in implantation, and from the tropoblast cells, the four extra embryonic membranes are formed. One is amnion, chorion, allantois, and yolk sac. And tropoblast cells nourish the embryo and helps in implantation. So when you look at the structure of blastocyst, some of the tropoblast cells are intimate contact with the inner cell mass or contact with the embryonal knob, or inner cell mass are called cells of Ruber. Okay, so look at here. These are the cells of Ruber, and these trophoblast cells are intimate contact with the contact with the inner cell mass. When it reaches the blastocyst, the zona pellucida disappears, and the blastula size increases in volume. It increases 0.15 mm to 0.30 mm, and the blastocyst stage is reached to 5 days after the fertilization. So soon after fertilization, or it moves towards the wall of the uterus in about sixth day after the fertilization. Sixth day after the fertilization, when it moves towards the wall of the uterus, the trophoblast cells secrete proteolytic enzymes or hydrolytic enzymes, and those hydrolytic enzymes which dissolves the uterine endometrium and creates the depression, and within the depression the blastocyst get buried by the process called implantation. So next, look at here. When you look at the structure of blastula, the exposed trophoblast cells stick to the endometrium at the embryonic pole. These are the exposed, when you look at these are the exposed trophoblast cells stick to the embryonic endometrium. Then during implantation, trophoblast cells at animal pole stick to the uterine wall, and it stimulates rapid cell division. It forms two layers. It forms two layer. One is syncytotrophoblast, and the second one is the cytotrophoblast. So after attaching to the uterine endometrium, the trophoblast cells differentiated into two layers. One is syncytotrophoblast, another one is cytotrophoblast. So what is the function? Syncytotrophoblast cells retain their cell membrane, forms a future amnion, whereas whereas cytotrophoblast cells, so trophoblast cells lose their cell membrane and appear multinucleated and release lytic enzymes that destroy the endometrium for implantation. So blastula gets buried into the uterine endometrium. So importance of implantation: it fixes and protects the embryo, provides nourishment to the embryo directly to the mother body, and the fate of soon after implantation, the blastula enters into the gastrulation process. So what is gastrulation? The single layered blastula is transformed into three layered three layer gastrula. So as a result of gastrulation, three germ layers are formed, that is outer ectoderm, inner endoderm, in between you find mesoderm. Soon after the formation of three germ layers, the two important processes takes place. One is histogenesis, another one is organogenesis. Histogenesis means the formation of tissues, whereas organogenesis means the formation of organs. So as a result of this, from these three germ layers, tissues and organs are derived. For example, from the ectoderm, epidermis, pituitary gland, tooth enamel, nervous system, conjunctive, cornea, lens, retina, hairs and nails, adrenal medulla, and stomatium. So these are the structures which are derived from ectoderm. Let us move on to the endoderm, endoderm or mesoderms. So from the mesoderm, dermis of the skin, muscular tissue, circulatory system, lymphatic vessels, tooth dentin, kidney, adrenal cortex. Look at here. Adrenal medulla is derived from ectoderm. Adrenal cortex is derived from mesoderm. Testes and ovary and coelom. So xylem is derived from mesoderm, middle edge. Okay. Then lastly we'll move on to the endoderm. So derivatives of endoderm: epithelium of midgut, digestive glands such as liver, pancreas, salivary glands, tongue, tonsils, respiratory system, thyroid, parathyroid, thymus gland, urinary bladder. So kidneys are derived from mesoderm, whereas urinary bladder and epithelium of urethra is derived from endoderm. In case of female reproductive system, vagina, vestibule is derived from endoderm. So this completes implantation and the derivatives of three germ layers. Thank you.